antipsychotic drugs, they can block the D2 receptor, and the affinity to the D2 receptor correlate very well with the clinical dose, which means that if the drug have a higher binding affinity to the D2 receptor, then you can use a lower concentration in the patient, lower dose. So that's good correlation. So that strengthens the idea that the D2 receptor is a major contributor to the disease. And then also you learned it in the Parkinson's disease treatment that some of the drugs that can increase dopaminergic activities, they actually can induce symptoms that look like psychosis. So all this, also together with some other relatively minor corroborative evidence, suggests that the dopaminergic active system is very important. And there seems to be too much dopamine in the brain. Now, Dr. Jen also proposed that in the mesocortical system, maybe there are too little dopamine. That concept is somewhat controversial. It's not universally accepted, okay? So, but the idea that too much dopamine is involved in the psychosis symptoms, that is very well accepted by the community. As Dr. Emmett already mentioned that in this area of drugs, there are a lot of controversies, and there are papers that are directly contradictory to each other. And then also textbooks are not the same. There are two textbooks. One will say that, oh, this drug has a lot of sedative effect. The other textbook said that, no, it doesn't. So it all depends on which particular studies one would quote, okay? So, so be mindful of that. Okay, so this graph shows you the correlation between how much clinical dose of a particular antipsychotic drug versus the binding affinity to the D2 receptor. For the drugs that has a high affinity, then the concentration or the dose you need to use to treat the patient become less. So you can see that this is really a very, very beautiful correlation. So that really strongly suggests that blocking the D2 receptor can help to treat or to manage the symptoms. And if you do the same thing for the D1 receptor versus the clinical dose, you can see that there's absolutely no correlation, okay? So blocking the D1 receptor does not seem to help the patients. And blocking the D2 really correlate very, very well. So the blocking D2 is a wonderful, important concept for treating schizophrenia. And Dr. Jen already mentioned that there are 4 major pathways, right? The nigrostriatal pathway for dopamine is starting from the substantia nigra and projecting to the striatum, and that is related to fine-tuning the motion. So that is the key pathway that is involved in Parkinson's disease, right? Then the tuberoinfundibular pathway is initiating in the hypothalamus that goes down to the top part of the pituitary, and it will release dopamine. And in early days, dopamine in the pituitary is also called prolactin inhibiting hormone. Before it is identified to be simply dopamine. So that once it's released in the top part of the pituitary, it can travel down to the lower part of the pituitary to control prolactin release, to inhibit prolactin release. So it is an inhibitory factor for prolactin release. Okay, so that's very important. And then we talk about the mesolimbic and mesocortical systems that is involved in psychosis. But it's also involved in emotion, reward, memory, and also motor functions too. So you can imagine that if you mess with the dopaminergic system, you are going to mess a lot of things. Therefore, we try not to do too much about it, and in psychosis, it's such a very debilitating disease. So even though with all these potential bad things that might happen, we still want to treat it, right? Okay. As I said, as you already heard from Dr. Ahmed as well as Dr. Jan, the dopamine hypothesis is not the whole story. There are other pieces of the story that do not directly fit with the D2 receptor. It's the only contributing factors. Because as Dr. Jan already talked about, in some area, the D2 receptor is actually lower than normal. So how can you just say that too much dopamine is bad, right? So, and then also D1 receptors also decrease in some of the regions. So there are concerns that maybe in other cases or in parts of the symptoms of psychosis, there are not enough dopaminergic activity too. But also Dr. Jan already mentioned that ketamine as well as phencyclidine, these two drugs can block the NMDA receptor. NMDA receptor is a subtype of glutamate receptor. By blocking this NMDA receptor, they can also produce schizophrenic symptoms. So there is the idea that glutamate receptor is involved. So the idea is that how maybe if we can increase the activity of glutamatergic neurons, we may be able to overcome some of that. But as Dr. Jan said, that many of those clinical trials failed. One major reason is the side effects, because glutamate is involved in so many vital functions in the brain. Or the fact that you are paying attention, you are learning, you are alert, these are all the result of glutamate. But too much glutamate kills neurons too. So therefore, if you inject monosodium glutamate, many Orientals put it in the food, into the brain, you can kill brain cells. So this, even though this concept is probable, but there are not too many active drug discovery programs that are simply aiming to enhance the glutamatergic activity. And then also there is the serotonin receptor hypothesis based on the fact that serotonergic neurons, the distribution of them are very closely related to the same region of the dopaminergic neurons. Actually, serotonergic neurons also seems to be more broadly spaced, so they actually innervate more areas. But many of them are actually co-localized with the dopaminergic neurons. And then blood serotonin levels is higher in schizophrenic patients with the abnormal brain scan. With that, as well as other evidence that the serotonin receptors also proposed too much serotonin may contribute to schizophrenia. Okay? So dopamine, serotonin. So this is simply show you that the yellow is dopamine, so this is the distribution of dopamine, and then the red is the serotoninergic distribution. And this is a raphe nucleus.